Assalamualaikum and hi everyone. My name is Alisa and today I will be presenting the introduction and overview for chapter 3, which is apparatus, techniques and reagents. A chemistry laboratory is a controlled environment where scientific experiments are carried out. It allows us to study different substances, chemical reactions, and their properties in a safe and structured way. In this space, students and scientists can observe real chemical changes and understand theories more clearly through practical work. In laboratory experiments, apparatus, techniques, and reagents all play very important roles. Apparatus refers to the tools and equipment used during experiments, such as beakers, burettes, and pipettes. For example, a pipette is used to measure and transfer small amounts of liquid accurately. Techniques refers to correct methods used when performing experiments. Using proper techniques is important because it ensures accuracy, consistency, and reliable results. Even a small mistake in the technique can affect the final outcome of an experiment. Reagents are the chemical substances used in reactions. They are carefully selected depending on the type of experiment being conducted. Without the correct reagents, a reaction may not occur or may give incorrect results. Chemistry laboratory work itself refers to practical experiments that involve the use of chemical apparatus and scientific procedures. It focuses on observation, measurement, recording data, and analysis. Through laboratory work, we are able to test and confirm scientific theories that we learn in class. Safety is also important. It is a very important aspect in the laboratory. We must always wear proper personal protective equipment such as lab coats, safety goggles, and gloves. In addition, we must follow all instructions carefully, handle chemicals responsibly, and avoid any unsafe behavior in the lab. It is also important to use emergency procedures such as the use of eye wash station and fire safety equipment.
Now, I will explain about chemistry laboratory apparatus. Different apparatus are designed for different purposes such as measuring, mixing, heating, and transferring chemicals. First, apparatus used for measuring liquids include the measuring cylinder, pipette, and burette. A measuring cylinder is used to measure the volume of liquid. It provides moderate accuracy and is commonly used in many laboratory experiments. Next, the pipette is used to transfer small amounts of liquid accurately from one container to another. It is commonly used when precise measurement is needed. Another important apparatus is the burette. The burette is mainly used in titration experiments because it can release liquids very accurately and slowly. Besides measuring apparatus, there are also apparatus used for mixing and heating substance. A beaker is commonly used to hold, mix, and heat chemicals. It is one of the most basic apparatus in the laboratory. The conical flask is used for mixing solution. Its narrow help necks prevents spilling while shaking the solution. Next, the test tube is used for small-scale chemicals reaction and testing substance. The Bunsen burner is used to heat chemicals during experiment.
Now, let's see the application. You might think lab equipment is only for mad scientists, but you use version of them every day. Take a beaker or a measuring cylinder in your kitchen. That's just your standard liquid measuring cup used to bake a cake or measure water for rice. What about a pipette or burette? If ever used a precise medicine proper for a baby, or seen a hospital IV drip perfectly regulate medicine drop by drop, you have seen this tool in action. And that conical flask we use to swirl chemical without spilling is the exact same design concept as a wine decanter or a fancy cocktail shaker.
I will explain about chemistry technique that used in lab. We have 4 techniques, which is titration, filtration, distillation, and chromatography. The first one is titration. Titration is technique that used to find concentration of unknown solution by reacting it with solution that have known concentration. Basically, we use chemical reaction between two solutions to determine the amount of one substance in sample. For the process, the solution with non-concentration is placed inside a burette, while the solution with unknown concentration is placed on the flask. The solution from the burette is slowly added into the flask until it reaches the endpoint, which can be seen through a color change using indicator. After that, the volume can be used to calculate and find the concentration of unknown solution. And next we have filtration. Filtration is a simple technique used to separate solid and liquid. It works because filter paper can block solid particle but still allow the liquid to pass through. During the process, the mixture is poured into a funnel with filter paper around it. The liquid that passes through is called filtrate, while the solid that stays on filter paper is called residue. Next, we have distillation. Distillation is used when we want to separate substances that have different boiling point. The main idea that the substance with lower boiling point will evaporate first. In the process, the mixture is heated until one component changes into a vapor. Then, the vapor moves through a condenser where it cools down and turns back into liquid. Lastly, we have chromatography. Chromatography is a technique used to separate different components in mixture. This happens because each component has different attraction towards stationary phase and mobile phase. For example, in chromatography paper, the sample is placed on paper and solvent moves to work through the paper. Different substances will travel different speed, causing them to separate into different spots. Now, let's talk about the applications. For example, filtration. Every morning, when you make drip coffee, the paper filter acts as the residue barrier, trapping the coffee ground while letting the liquid filtrate pass through. Then there is distillation, which boils and condenses liquid based on boiling points. This technique is literally how we turn salty seawater into fresh drinking water, and how luxury perfume extract beautiful essential oils from plants. We also have titration to test chemical concentration. Lifeguard also use a mini version of this to check if pool water is safe, and chromatography, which forensic scientists use to separate ink or blood sample to solve crimes.
Assalamu alaikum and good day everyone. My name is Muhammad Firdaus and today I will be presenting part 4 of chapter 3, which focuses on chemical reagents and the conclusion. In this presentation, I will explain 4 commonly used chemical reagents in laboratory experiments, which are hydrochloric acid, sodium hydroxide, phenolphthalein, and Benedict solution. I will also discuss their functions, reactions, safety precautions, and conclude by explaining the importance of apparatus, techniques, reagents, and proper laboratory procedures. The first reagent is hydrochloric acid or HCl. Hydrochloric acid is a strong acid that is commonly used in chemistry laboratories. It is mainly used in acid-based reactions and titration experiments. Its function is to react with alkalis to produce salt and water through a neutralization reaction. For example, hydrochloric acid reacts with sodium hydroxide according to the following equation. HCl plus NOH produces NaCl plus H2O. These reactions form sodium chloride and water. Since hydrochloric acid is highly corrosive, it must be handled carefully. We should always wear safety goggles, gloves, and a laboratory coat. Direct contact with the skin or eyes should be avoided, and any spills should be cleaned immediately according to laboratory safety procedures. The second reagent is sodium hydroxide, also known as NaOH. Sodium hydroxide is a strong alkali that is widely used in neutralization reactions and titration experiments. Its main function is to react with acids to produce salt and water. For example, sodium hydroxide reacts with hydrochloric acid to form sodium chloride and water. Although sodium hydroxide is commonly used in laboratories, it is highly corrosive and can cause burns if it comes into contact with the skin or eyes. Therefore, we should wear gloves, safety goggles, and a laboratory coat while handling it, and wash any affected areas immediately with plenty of water if accidental contact occurs. The third reagent is phenolphthalein. Phenolphthalein is an acid-based indicator that is mainly used in titration experiments. Its function is to indicate the endpoint of a neutralization reaction through a color change. In acidic solutions, phenolphthalein is colorless. However, in alkaline solutions, it changes to a pink color. This color change helps determine when the acid and alkali have completely reacted. Although phenolphthalein is only used in small amounts, it should still be handled carefully. Avoid direct contact with the solution and follow all laboratory safety instructions. The last reagent is Benedict solution. Benedict solution is commonly used to test for reducing sugars such as glucose. During the experiment, Benedict solution is mixed with the sample and heated. If reducing sugar is present, the blue solution changes color to green, yellow, orange, or brick red depending on the concentration of sugar. When performing this experiment, test tubes should be heated carefully using the correct laboratory techniques, and contact with the reagent should be avoided.
In the lab, we use hydrochloric acid, but did you know your stomach naturally produces it to digest your lunch? We also use sodium hydroxide, a strong base. In your house, that's the active ingredient in heavy-duty drain openers, and it's even used to make everyday bar soap. We also use indicators like phenolphthalein, which turns bright pink in a base. The secret of behind cold disappearing inks. Benedict solution, which changes color to detect sugars, was historically used to test for diabetes. To wrap it all up, understanding apparatus, techniques, and reagents isn't just about passing a chemistry quiz. It's the foundation of how humanity mass produces safe food, creates life-saving medicines, and keeps our water clean.
In conclusion, laboratory apparatus, techniques, and reagents are all essential components of chemistry experiments. Laboratory apparatus allows scientists to measure, transfer, and contain chemicals accurately. Laboratory techniques ensure that experiments are carried out correctly, systematically, and safely. Chemical reagents make it possible to produce and observe chemical reactions, allowing scientists to collect accurate and reliable data. Following proper laboratory procedures is equally important because it reduces the risk of accidents, protects laboratory users, and improves the accuracy and reliability of experimental results.